because I usually run out of time, and I want to make sure I acknowledge everyone involved in this work. So the real hero here is Jin Young Kang, and you'll see more of that later. Also Eliza Llewellyn, who was a research assistant working with Jin Young. James Chen, the sort of cryo-EM guru in the lab. We collaborate a lot with Brian Chait and Dom in Brian's lab, who are spectacular mass spectrometrists. Carol mentioned Liz here. And a relatively new student on the project, Joshua Brewer, who contributed to this work, but he's also carrying it in new directions. So Evelyn, happy birthday. We actually met once for about 2 minutes. I saw your lecture when you received the Wiley Prize at Rockefeller. I think it was 2015. But I've known of you and of your work for a very long time. I actually often, I try to encourage students, and in my own writing, I try to, reference original manuscripts rather than, subsequent reviews and stuff. Try not to be lazy. And what I find myself, especially in our work on MFD, is that it always comes back to referencing your papers. And as Steve Elledge alluded to, at the time you were doing these experiments, there wasn't a lot of information to go on. And so it's really amazing how much you could figure out by looking at, bacterial plates, basically, as far as I can tell. And, Evelyn was really very far ahead of her time, as we've also heard people acknowledge, not only scientifically, but also from the perspective of civil rights. And President Holloway alluded to this earlier. I'll go over this quite quickly. At the time when Evelyn was an undergrad at NYU, schools like NYU had sports teams that had Black players on them. But when they played schools from the South or segregated schools, they had a sort of an agreement that the Black players would not play. And Evelyn and a group of 6 other students mounted a vigorous protest when NYU was going to play Missouri, and NYU had a star football player who was Black. His name was Bates, Leonard Bates, and he wasn't going to be allowed to play in the game. And this protest actually resulted in these 7 students, called the Bates 7, getting suspended. And so Evelyn was not able to complete her graduate degree on time. And I think that led to her not going to graduate school at Columbia, and instead went to Cold Spring Harbor. And I mentioned this is ahead of her time because this was actually 6 years before Jackie Robinson played in Major League Baseball. This was 14 years before Rosa Parks, and more than 2 decades before Martin Luther King, his famous speech. So Evelyn went to Cold Spring Harbor, and we've heard a lot about her prescient and amazing work characterizing the SOS response. But I'm going to talk a little bit about something that hasn't been mentioned too much, and that's MFD, mutation frequency decline. So Carol just outlined this a little bit. Mutation frequency decline is defined actually in a paper from Evelyn as the rapid and irreversible decrease in the frequency of certain UV-induced prototrophic mutations that occurred when protein synthesis was briefly inhibited immediately after UV irradiation. So I'm not a genius like Evelyn or Carol. And so, when I read that, my first reaction is, huh? And here's an example of the data from one of Evelyn's papers. This decrease in mutations, but she discovered this gene MFD when it's the function is knocked out. You see this phenotype. But actually, so this sort of description is really hard for me to understand. A lot of it seems sort of ad hoc. Why would you do it this way? Why would you inhibit protein synthesis? But actually, I encourage everyone to read this paper here. that's sort of a perspective much later that was written by Evelyn that really sort of explains a lot of this, and a lot of this makes sense once you, at least for me, when I read through this paper. But I don't have time to go through all this right now. As Carol also alluded to, around this time, Phil Hannawalt discovered this phenomenon of that we now call transcription-coupled repair. And so that is the much more rapid repair on the transcribed strand of actively transcribed genes than either uninduced genes or the other strand, the non-transcribed strand, or we call the non-template strand. And I put these boxes around because I think for a long time, these two phenomena sort of seem unrelated. And Carol mentioned this paper from Aziz Sankar, Evelyn, and Chris Selby that really showed that in bacteria, the protein involved in mediating this response, this transcription-coupled repair response, is this MFD protein. So sort of unifying all these things together into a way that's starting to make sense. So subsequent work from many, mostly from Chris Selby and Aziz Sankar and others, led to a sort of model for how this was working in E. coli. So in this example from one of Selby and Sankar's papers, the RNA polymerase is transcribing DNA that has a lesion on the non-template strand. And what happens is polymerase just happily transcribes through. But if the lesion is on the template strand, the strand that the RNA polymerase is reading, certain types of lesions cause polymerase to stall. It can't read through the lesion. The most common is a pyrimidine dimer. And so this is a disaster on many levels. It blocks synthesis, further transcription of the gene. It causes all kinds of other problems. And furthermore, the RNA polymerase stuck here, the actual damage, as we know from work of Patrick Kramer, Dong Wang, and others, actually is right inside the RNA polymerase, almost to the active site when it causes the polymerase to stall. So this damage is buried inside the RNA polymerase, and it's not accessible to repair factors. And it was known early on that this stalling of the RNA polymerase inhibits repair. So this MFD protein, that's also called transcription repair coupling factor, comes into action. And in a process that involves ATP hydrolysis, it removes the polymerase from the site. And then subsequently, not only does it relieve the inhibition of repair, it also stays and recruits nucleotide excision repair proteins. So sort of this sort of shortcut entry into the nucleotide excision repair pathway. And that's how transcription-coupled repair stimulates repair of the transcribed strand over the non-transcribed strand, the model anyway. So actually many years later, a really talented student in my lab, Alexandra Diaconescu, who's now a professor at Brown University, used X-ray crystallography to determine the structure of this MFD protein. This is the structure she determined. And one of the remarkable things about this structure is that it's basically a series of protein domains linked by sometimes extremely long, like 30, 40 amino acid linkers. So one of these domains actually looks like one of the NER proteins, this UVRB homology module. This is the connection with recruiting the UVRA protein to the damage. There's a D3 domain that we don't know what it does. It's not present in all MFDs. There's a translocation module that's basically a RecG-like ATPase domain that serves not as a helicase, that's often called, these are often called helicase motifs, but MFD is actually a double-stranded DNA translocase. There's a RID domain that stands for RNA polymerase interacting domain that is known to be the point of interaction between the RNA polymerase and MFD. And then finally, there's this C-terminal D7 domain that really serves to sort of you'll see here in a minute, interact with the UVRB, the N-terminal part of the protein, and sort of hold it into this sort of what we now know as an inactive conformation. The next thing that happened really was a couple of remarkable papers from Jeff Roberts' lab that really highlighted more the biochemical or the biophysical function of MFD. And what Jeff's lab showed is that this is a schematic of a backtracked RNA polymerase transcription complex. The polymerase is moved backwards. And when that happens, the RNA transcript, which is shown in red, reverse threads through the complex. And here's the 3' sort of single strand fragment. So these complexes are inactive because the 3' end of the transcript is not in the active site of the polymerase. So what Jeff showed is that MFD engages with these complexes. It requires about 20, 30 base pairs of double-stranded DNA upstream of the RNA polymerase. And then in a process that involves ATP hydrolysis, MFD uses its translocation activity to basically push the polymerase forward. So re-engaging the 3' end of the transcript in the active site. And then the polymerase can, elongation activity of the polymerase can take off and MFD falls away. But if there's a lesion in the DNA that the polymerase cannot travel through, the polymerase stalls at the lesion. It may backtrack. MFD can engage and push it forward just like it did before. But now it can't keep pushing polymerase forward. The elongation activity of polymerase cannot take over because of this lesion. But MFD doesn't know that. It just keeps doing its thing. So it's hydrolyzing ATP, translocating on the DNA. And the model is that instead of pushing polymerase forward, now it's basically pulling the DNA and torquing the DNA out the back of the RNA polymerase. And what happens is this causes the transcription bubble to collapse, probably from the upstream end. And eventually this causes complete collapse of the bubble, release of the RNA, and release of the RNA polymerase. And then really unexpectedly from work of Terence Strick and Nigel Savory, who are both here, what Terence found is that once the polymerase is removed from the DNA, it stays associated with MFD, and MFD, in the absence of any other factors, just sort of slowly translocates on the DNA in the downstream direction. And what he actually showed is that this translocation can happen, is extremely processive. So these are traces that are on pieces of DNA that are 3 kb long. Now, this X-ray crystal structure of MFD, I showed you here. Here's another view of it. And I mentioned already in this apo form that we call, all these cool activities of MFD are really strongly repressed. So MFD doesn't really bind polymerase very well. It doesn't bind DNA. It doesn't hydrolyze ATP. It doesn't translocate on DNA. So all of these activities require, activities are unleashed by engagement with the elongation complex. And this is expected to be accompanied by really profound conformational changes in MFD. So I showed you how MFD is sort of a bunch of domains linked by long linkers. And actually Alex proposed in her paper in 2006 that MFD was sort of built for conformation changes. Oops. So we wanted to see what these conformation changes were. We worked for actually a long time using X-ray crystallography and getting nowhere. And then, as many of you know, the cryo-EM resolution revolution happened, and this really, I'll show you in a minute why this allows us to do this work. So we Jin Young set out to use cryo-EM. I'm having trouble because, okay. To really see MFD in action. And so what she used is this nucleic acid scaffold to assemble elongation complexes of RNA polymerase. And the key points about this scaffold are it's designed to be a post-translocated elongation complex. there's a part of the transcription bubble that's non-complementary. So even if MFD latches on and starts doing its thing to remove the polymerase, it can't really collapse the transcription bubble because it's non-complementary. And so we have a chance of catching it in cryo-EM, even though we're not trapping it or inhibiting it or anything like that. And in addition, this scaffold, as you can clearly see, has a large segment of double-stranded DNA upstream of where the RNA polymerase will be. And this is required for MFD to load on. And so just some biochemical analysis. Here's a native gel. The top panel is Kumasi stain and the bottom panel is stained for DNA. And here's what you see if you run MFD. Here's if you run RNAP. The polymerase tends to oligomerize and you get kind of a smear. If we assemble this elongation complex with the RNA polymerase, you get this band. And if you just take this elongation complex and add MFD, you just get an MFD band and an elongation complex band, nothing really happens. But as soon as you add ATP, you get a new band. And Eliza showed, she cut out this band and ran it in an SDS gel, and the band contains RNA polymerase and it also contains MFD and it contains DNA. So this is the thing that we want to see. We also use with Brian and Dom native mass spectrometry, and we also show the generation of this stable complex that's the EC stably bound to the MFD in the presence of ATP. Okay, so normally in a talk like this, I don't show this kind of detail of the cryo-EM, but this is really the key to the whole story. And that is sort of, we have MFD bound to this elongation complex in the presence of ATP. So MFD is really going through its ATP hydrolysis. It's doing its thing. It just can't knock off the polymerase because the transcription bubble is not complementary. So we knew going in that this sample was going to be very heterogeneous. And the real key to this whole thing was Jin Young's ability to go through all the sample, all the particles that she found, and sort out all the different conformational states of the complex that are present. And what we ended up deciding was that there were 7 pretty well represented complexes. They're shown here in this sort of classification pipeline. And what I'd like to show you now are these 7 structures. So just to orient everyone, this is a view of the elongation complex of RNA polymerase that it was actually determined by Jin Young in an earlier publication. The polymerase is like a big crab claw, but you're looking from the top. So the channel is in between this blue and pink subunit. The blue subunit is the beta subunit, pink one is beta prime. And these are transparent so you can see the nucleic acids inside. So this is the downstream DNA. This has not been transcribed yet. This is upstream DNA. It's already been transcribed. Here's the transcription bubble being held open inside the polymerase. Here's the active site of the polymerase, the 3' end of the RNA. The RNA transcript is colored red here, and this short part of the transcript is in an RNA DNA hybrid with the template strand of the DNA, the one that's being read. So here's the same view of the elongation complex of the RNA polymerase. You don't see the DNA because it's too short to stick out, but the upstream DNA is here, and you see MFD associated with using the same color coding that I was using before. And now these are the 7 structures that Jin Young isolated. They're all close to 4 angstroms. Most of them are much better than 4 angstroms, almost 3 angstrom resolution, which when this was done was pretty remarkable. So the thing about all these structures is the EC doesn't really change that much. It does go through some changes that I'll describe in a minute. What you'll notice is the RID, this magenta part, engaged with a part of the polymerase we call the beta protrusion. And this doesn't really change all of the structures. The RID is clamped onto the beta protrusion. But the rest of MFD is going through these sort of molecular contortions that I'll try to describe to you here. And I'll point out that among all these structures, if we align the RNA polymerase, we get RMSDs ranging from here. But if we align the MFD part, they're completely not the same molecule at all. So what I'd like to do is go through the different structures, kind of a movie. What did I do here? Okay. And you'll notice that as you go through these structures, the polymerase doesn't really change that much, and MFD is going through all kinds of contortions. Let's go through that again. And I'll try to explain a little bit about what some of these contortions are. This is all of the structures superimposed. So one thing we could see very clearly in the different structures, some of them had ATP in them, some of them had ADP. So we really were seeing MFD going through its nucleotide hydrolysis cycle. In the ATP structures, the gamma phosphate is engaged with the two domains, both domains of the translocation module, the yellow domain and the green domain. But in the ADP structures, the gamma phosphate's gone. You lose these interactions, and this green domain rotates, sort of loses contact and rotates away from the yellow domain. So if we line up all of our structures by the yellow domain and we compare the position of the other domain, you see there's a group of them that align with the ATP and another group of them that bunch together in the ADP conformation. And if you look at the rotation is about a 16-degree rotation. And if you look at the movement of the center of gravity between the green and the blue domain, it's 3.5 angstroms, which is basically a base pair. So we think this is the basis of a sort of inch-forming mechanism that I'd like to show here. That is, been presumed to work for other types of helicases and translocases. So in this case, the ADP conformation opens and then it binds ATP and closes again. And in that way, it holds on and lets go of different parts of the DNA and is able to walk along the DNA in this manner. Now we tried to show if the MFD is connected to the polymerase, which cannot move, it's still going to be going through this cycle. So instead of walking on the DNA, it's going to be pulling the DNA along with MFD being stationary. And that's the basis for sort of winding up the transcription bubble. I want to go through what we call the loading part of this pathway. This is actually the first structure. We're looking right down the axis of the upstream DNA. And you'll notice in the second structure, the two motor domains are completely on the opposite side of the DNA. They didn't, it looks like they could just sort of move over this way, but actually we know they went this way. So they had to go around the DNA like almost more than 180 degrees, because as they do so, they wrap a part of the yellow domain around the DNA. And I'll show you that here in this movie. So this is the elongation complex. Here comes MFD. It engages with the complex. Now it's bound to ATP. And what happens is the domains start walking on the DNA. And as they do that, you'll see this yellow helix called the relay helix that gets sort of dragged around the DNA. The relay helix. This is dragging the red domain with it. So the relay helix gets pulled apart and is sort of wrapped around like a leash around the DNA. And so in the end, you end up with MFD completely topologically wrapped around the DNA. And we think that's the basis for this processivity, this remarkable processivity of this walking that happens. Once this process is finished, this N-terminal domain rearranges, and now it exposes this part of the protein that can bind the NER components. And I think, I have no idea how much time I have. So maybe I'll skip that and go into the other part. So once MFD is loaded, it's exposed this part of the protein that can interact with the NER components. And then we think that it continues to hydrolyze ATP, but now it can't really walk forward on the DNA because the RNA polymerase, the translocation modules run into the RNA polymerase. And as it goes through this cycle, MFD imparts conformation changes on the RNA polymerase. One of these is opening of the clamp, almost a 20-degree opening of the clamp in one part of the cycle. Another part is the rotation of this other domain we call the lobe, that we also know is involved in destabilizing interactions with the DNA. And so you'll see this in another movie where we're done with the loading pathway. We're just going through the nucleotide hydrolysis pathway, where MFD is sort of impinging itself on the polymerase and causing two conformational changes in the polymerase that we'll highlight here by just getting rid of. So MFD is there. You see it opening the clamp, and at a different stage of the cycle, rotating the lobe. And both of these changes sort of loosen the grip of the RNA polymerase on the DNA, and we think help disrupt the complex in addition to winding up the transcription bubble. So I'm not going to go through how we worked out this pathway, but basically MFD engages with this stalled elongation complex, goes through this remarkable conformation changes that involve hydrolysis of a lot of ATP before it even gets to the point where it can start collapsing the transcription bubble, and then it has to go through this cycle probably many times before it actually can remove the RNA polymerase. So why have such an elaborate mechanism? It's because we know that from the work of Bob Lanik and many others that transcriptional pausing is an important regulatory mechanism in both bacteria and eukaryotes. So we don't want MFD engaging with a paused RNA polymerase and removing it, because a lot of these paused polymerases are supposed to be there. But actually in E. coli, at least, pause half-lives in vivo have been estimated. The His pause half-life, which is one of the longest lived pauses in vivo, was estimated to be between 1 to 2 seconds. And from Terrence's in vitro single molecule experiments, the mean lifetime of the MFD elongation complex displacement complex, so this whole process that I just described to you, between MFD engagement and RNAP displacement can take as long as 45 seconds. So the expected time required for MFD to find and displace an elongation complex is more than an order of magnitude longer than the longest lived transcriptional pauses. So we think the way MFD discriminates between complexes that it's supposed to remove and paused complexes it's not supposed to remove is kinetically. And this helps explain why overexpression of MFD is not toxic to cells. I thanked Josh, Liz, James, Eliza, and most importantly, Jin Young, who's not on this list because she's not in the lab anymore. She has her own position at the KASTE Institute in South Korea. Collaboration with Brian and Dom, and great collaboration and help from our cryo-EM center at Rockefeller, Mark Ibrahim and Johanna Sotiris. This has been a long-standing sort of great interaction, collaboration, discussions with Terrence and Nigel. And I didn't talk about any of this work today. It's really sort of just at the ground level. But Liz and I have started a collaboration with Hura also that we're very excited about that relates to this work on MFD. So thanks very much. I'll stop there.